Yes, sure, you will see that this is not a place to find salmon or a place to cultivate algae. These are really deep, dark environments found in our deep ocean. And the picture you see here is what we call a hydrothermal chimney. And the research that I will present today has been performed at the Center for Geobiology starting in 2007. And we now also have a Center for Deep Sea Research here in Bergen. And I will start with presenting some of the basic research that we do. And then I will try to convince you that there seems to be many interesting microorganisms, interesting biodiversity, biomolecules in these environments that can have relevance for biotechnology. And where do we find these hydrothermal vent systems? They are found, as I said, in our deep ocean, and it is really a big biosphere. And what is really important here is that they were discovered as late as 1977, and it's a really big biosphere that we know very little about. And this is maybe what you know from these vent fields. This is a black smoker, and the fluids that are emitted from these black smokers can have a temperature up to 400 degrees. And these environments have been sites for finding microorganisms that can live at really high temperatures. So the record holder at the moment lives at 122 degrees. So these are really extreme environments. And again, they are photosynthesis independent. There is no light here. And the microorganisms and the food webs are fueled by chemosynthesis, meaning that the microorganisms use, for instance, hydrogen, methane, and hydrogen sulfide as an electron donor, and they can respire with oxygen, sulfate, nitrate, and carbon dioxide, which is found in the cold surrounding seawater. So these microorganisms, they make organic carbon and do the CO2 fixation. And at a Center for Geobiology, we have discovered the Norwegian deep sea hydrothermal vent systems. They are located on the Arctic mid-ocean ridge, and we started out discovering the Jan Mayen vent fields that were the geologists that work at the Center for Geobiology. They discovered this in 2005. And since then, we have been working on mapping these hydrothermal vents field. And this is how we work. The geologists, they have, of course, an extensive knowledge on how to make maps. And that is what they do. They make maps. And then we know and they identify the hydrothermal vent fields. And then we biologists, we can go and take samples and we can, for instance, isolate novel microorganisms. One of the hydrothermal vent fields that we have studied the most is the so-called Loki's Castle vent field. This is located at 73 degrees north, at 2,300 meters below sea level. And we say that this hydrothermal vent field has a novel chemistry, meaning that it is very high in methane. And this is explained by the geologists that it's influenced by sediments coming from the Bear Island fan. So the geological reactions, the geochemistry, knowledge on that is really important. And of course, as you have learned from Phil earlier today, when you study these microbial communities, we want to use different omics technologies. So of course, we use omics technologies, we use microscopy, and for us it's really important to have the geochemistry of the system. And this is how we work. This is our ROV. So here we take a sample at 2,300 meters depth. This is a biofilm growing on a black smoker. And this sample was taken in 2009. And of course, when you see something like that, you want to know which microorganisms are living there. How do they look like? And this is how they look like. These are really big cells. And there are some smaller cells growing on these large filaments. And of course, we want to give a name on these microorganisms. And in this case, we have sulforovum, which is a group of epsilon protobacteria. And we also have Bacteroidetes, which you find in the human gut, for instance. And these are organotrophs. And when we look more in detail, we found a very thick sheet surrounding these cells. And when you see something like that, you want to learn more about it. We have been working together with different groups trying to find out what this polymer is made of. We don't know yet, but we know that this is actually a very thermostable polymer that can be boiled at high temperatures. So this is maybe a novel biopolymer formed by chemosynthetic microorganisms, something similar to what you can find of biopolymers produced by photosynthetic microorganisms or plants. And for us, it's of course important to understand the ecosystem, to understand how the transcriptome and proteome and how the community utilize the chemistry. And for us, it's really important then to have knowledge about which enzymes can use the different substrates that we see in the vent field. And this we have done. We have identified enzymes that can use, that can catalyze oxidation of hydrogen and hydrogen sulfide. And we know that this is done by these microorganisms. But when you go to these environments, of course, the map is important. And when we see that at Loki's Castle, we have these two sites of high temperature venting. But it's not always in these environments that you see so extreme environments. You can go and you can find more gentle environments where the environmental parameters are more moderate. So the temperature can be lower and the chemistry can be modified. And in this case, it's important here that in the fluids that come through these other kinds of chimneys, hydrogen is not present. And this is how it looks. These beautiful microbial mats are sitting here and fluids are venting through. And this is not a black smoker, this is called a barite chimney. And this is what we see, a completely different organization of the biofilm. Here we have single cells, we have organic polymer, and we have minerals being precipitated on the polymer being formed. And this is really an example of how geological processes work together with biological processes. And we have, of course, a name of the microorganisms. This is a different group of these epsilon protobacteria called sulforamonas. And as you may remember, there was no hydrogen coming through these chimneys. And when we looked into the metaomics data, we couldn't see any hydrogenases. And now we are inside the chimneys, and here we have found some beautiful long chains of what is called anaerobic methanotrophic archaea that can feed on the methane inside the chimneys. And at the moment, we use metagenomics to try to find out how their role in the community and how they interact with other microorganisms. And maybe a point here is actually when we started out in 2007, the technology, the sequencing technology, didn't allow us to do metagenomics of all of the samples that we collected from these systems. Because it required to do metagenomics and to use the omics technologies, we really need to have high biomass. So when we worked here, we started, of course, out with the biofilms. There we could get out microgram of DNA, protein, and even RNA. It is much more difficult to work with chimney materials. It's like taking DNA out of stones. So that's what we are faced with, low cell numbers and low yield of DNA. So with the new sequencing technologies where we can feed in much less DNA, we can now start to do metagenomics on samples that we couldn't do before. At the same time as we have been building our Center for Geobiology, we have also been involved in some biotech projects, the NorSAM-D project, for instance, in starting up 2012, and we are also part of 2 large Horizon 2020 projects. And what is interesting in these projects are, of course, that we find that organisms that can live at high temperatures. And that is because if you go to a biorefinery, often the temperature is very high. And you want to have enzymes that can tolerate high temperatures. And that is what we can find. Of course, if a microorganisms can live at 122 degrees in the deep ocean, they need to produce enzymes that can tolerate these high temperatures. And we have used the technology that we have developed during our Center for Geobiology period to build a biodiscovery pipeline where we have built a huge database and a gene catalog of encoding diverse enzymes. And this database and our enzymes from the Norwegian deep sea hydrothermal vents, we explore the use of these enzymes in different value chains. Marine value chains and green, and also food processing and molecular biology. And I will start with our Virus X project, then we go to the viruses. And this is a project starting in 2016. Arctic Science in Tromsø are also involved in it. And this is a project where we do viral metagenomics. And of course, this is, when you go out in nature, maybe viruses are the biological entities that we know the least about. But they are really abundant. And this is a really unexplored sequence space. And we say that this project is a journey into this unexplored territory. And you all know that viruses, they need a host cell to reproduce. That means that they have enzymes that have to catalyze these reactions. And these enzymes can be very important for biotechnology. And we know from only a few E. coli viruses that there are a number of enzymes that are really valuable. And one of these, for instance, is reverse transcriptase. But since there are so few viruses known, the sequences of viruses known, there should be a huge potential to find new, novel enzymes for molecular biology. And of course, we are not doing all in this part of these projects. Our role has been to develop a special system for taking large fluid samples at the seafloor. So we have developed this large water collector, because when you work with viruses and viromes, you need large volumes. So we have developed this so that we can pump in 50 liters of fluids, hydrothermal fluids, at 2,400 meters depth. But also there are other researchers at University of Bergen involved in this research. There is a long and big expertise in doing marine microbiology, and with a focus on virus research. And we have an expert on virums in our university. So she is Professor Rodanne Sander. She has protocols on how to get DNA out from virus and to sequence these. But again, when we do the sequencing here, it's really advanced to do the annotation of viruses. So this is not something that we are doing in Bergen. This is, of course, specialized experts in the consortium that are doing the annotation. And then to the INMARO project. So here we work on finding enzymes for food processing. And this just illustrates how we are working. We isolate microorganisms. We find which substrates they can utilize. We sequence the genome. And in some of the project, we also express the proteins and characterize the proteins. And then to just briefly mention the work that we are doing in NorSIMD. This is a project, Biotech 2021 project, and it's led by Vincent, starting up in 2012. And you have heard, of course, a lot of the work that they are doing on lignocellulose. But the aim of this project was to develop enzyme technology for processing of marine biomasses, or Norwegian biomasses, sorry. And we wanted to use Norwegian biodiversity to look for enzymes. And in this project, our role was to go to the deep sea hydrothermal vents and do metagenomics and to provide those data and collaborate with other researchers, the whole consortium, feeding sequences in the blue and the green pipeline. And of course, we have learned a lot about how how important it is to make new products from fish waste. And if you want to learn more about the enzymes for use of that, you can talk with Gro Berger and her team at Uni Research. I also found it very interesting because, of course, I didn't know that when we started, how many products you can get from wood and not only bioethanol. And of course, how important it is to find new enzymes to make new products. But our major role here was, as I said, to find new enzymes. But that is not so easy when you go down, as I have told you. These ecosystems, they are based on chemosynthesis, where hydrogen, methane, and hydrogen sulfide feed the microorganisms. And then you can question if this is the right place to go and look for enzymes that can be used for the degradation of complex organic compounds like wood and fish byproducts. And this was something that we wanted to see if we could do something with. And in that work, we actually wanted to feed the microorganisms with material from our industrial partners so that we could get the community to use these complex organic compounds instead of the gases, so that we could increase the fraction of relevant enzymes. And in that work, we developed these in situ chambers, which we filled with material that we obtained from the industry. We incubate these in situ chambers for 1 year on the seafloor, and here we collect some of them. And we have also developed a technology where we can measure the temperature. We can follow the in situ temperature during the incubation. So now we can see if in the different chambers, if the temperature is kept at high temperature during our incubations. And this is really important. If you want to look for enzymes operating at a certain temperature, you need to know which temperature you incubate under. And as I said, the technology, the sequencing technology has of course been very important. When we started out with sequencing our biofilms, we got some 10,000s of open reading frames using the 454 sequencing technologies. This has, of course, increased enormously when we sequence using the Illumina technology. And from a single chamber here, we can reconstruct 100 genomes. And this is now the stage where we are. We have a Venzyme database where we have over 4 million open reading frames. And this is from organisms that can live at very different temperatures. It's a biodiversity that is not present at NCBI. A lot of the biodiversity has never been seen before. So it's a really novel biodiversity. And it's also a really novel biochemistry, I would say. So this is, if you want to, I think that this is really a place to look for biochemical dark matter, microbial dark matter. And I think that since many of these organisms also are close to the root of the tree of life, it could also be very interesting to look into these organisms to see how different enzyme families have evolved. And of course, it's a really nice database to look for new enzymes in new value chains or existing value chains, but it's also challenging to work with this biodiversity. We have a lot of archaea, and this is organisms of the third domain of life, and sometimes it can be a bit more challenging to work, even though they usually function or expression can be expressed in E. coli, sometimes you cannot. It's also a data set and also an environment and that we can now know so much about, that we can see that we can do modeling on different levels. The modeling can be on ecosystem. We have a lot of ecosystem knowledge now. We have isolated single microorganisms from these environments, and we have the huge database of enzymes. So I think that we have now we have reached a level where we can go into the more digital world, and we have so huge data sets that we need new ways of analyzing our data sets. So, and then thank you to my team and the funding.
Thank you very much, and we have time for question. I think that this you presented here a truly transdisciplinary project that's really in the spirit of this DigitalLife project. It was hand here, yes.
Thank you, Ida. It was a really nice talk. There's one question I have. The bacterial communities in the Arctic thermal vents, how much do they differ from the bacterial communities in the thermal vents regions in the southern hemisphere?
That depends on where you look in the vent system. I think when you go and the biofilms, the biodiversity that we observe in the biofilms, that biodiversity is really typical biodiversity for hydrothermal vent systems. Then there is much less knowledge actually worldwide on what is present in the black smoker walls in the warm sites, because that is something that has been difficult to get access to, as I said, because of also the requirement for higher concentrations of DNA. We see that some data sets, some publications now are coming where they have used metagenomics to explore the microbial communities here. That makes it possible for us to compare with what we have in our vent systems.
Yeah, thanks for a very interesting talk. This looks very promising. I'm a sociologist of science, and we're looking before at processes of bioprospecting in other contexts. And right now it is quite interesting, the debate that is coming up on the Convention on Biological Diversity and the Cartagena Protocols on property rights, biodiversity, digital biodiversity, and so on. So I'm just wondering about what is your position on this in this project?
This we have been exploring in our project, but there is also now a new proposition coming for Norway about how we will deal with our biodiversity. At the moment, it seems that there is no requirement for rules on benefit sharing, but we have to register and apply for taking out material for bioprospecting.
I have one question for you. I was wondering on the status of the data. Will that be accessible, or if it will be when?
It will. We hope that it will get more and more accessible, but it's a big job to sort and to publish and to reconstruct the genomes, to analyze the genomes, and place that in those genomes in the tree of life doing phylogenomics. But we will, of course, continuously try to publish, and these data will, of course, be available.
Yes. Are there last question? Yes. Please go.
Thank you, Ida. Very nice presentation. I'm sort of relieved to hear that all presenters point to recombinant expression as a bottleneck in science. On the other hand, I'm becoming stressed because we're not, as a group, we cannot solve these problems that are continuously coming up with recombinant expression. Do you think the biodiversity you're seeing here, the enzymes in these microbes, do they require a special protocol for recombinant expression?
I think that is a good question, of course. As I said, I think we have a very most of them, or many of them, will be expressed in E. coli. But maybe in some and that's the first option. I mean, for basic research, that's the first choice. Of course, if you want to do more advanced expression for biotech, you may not use E. coli as the host. In some cases, but there are other that maybe know more about that than me. But I think that I think it's really difficult to know, because sometimes it's so difficult to know why an enzyme is not expressed. And what can we see in these proteins that can, why we not, I don't know, maybe we need in some cases some thermophilic hosts. That is something that is being developed by, and has been developed.
I'm asking myself the same question.
It's a good question. Okay, thank you very much, Ida. And here we have a small gift for you as well. Thank you.